Thank you, and it's a pleasure to have the opportunity to give an update on Miromatrix, where we are in our pipeline, where we are as we bring some of our key assets forward as well. So at Miromatrix, we're really driven by our mission, and our mission is to bioengineer transplantable organs to eliminate the transplant wait list. As we look at this field, we look at this sector, tremendous market opportunity here, or I would say patient need. There's about 120,000 patients in the US today waiting for an organ. 95% of those patients are either waiting for a liver or a kidney transplant. About 70,000 patients this year will go to their physician, be pre-screened, but not have access to life-saving technology. That's what we're looking to change. I'll talk about our technology, which has got tremendous differentiation in our platform of our ability to be able to bioengineer organs, as well as our pipeline. What have we done? What are our lead assets? And what is the timing for some of those as we continue to move forward? Through our journey, we've attracted key strategic investors, including DaVita, Baxter, and CareDx, as well as we have great scientific and medical collaborations with Mayo Clinic, Mount Sinai, and Texas Heart. Here's a quick video just to introduce you to the technology, and then we can dive a little bit deeper into that. If you could play that. Our patented perfusion decellularization technology starts with a functional organ from a pig that would normally be discarded. Next, we remove all the organ cells by first cannulating the organ and then flushing it with a simple solution that only removes the cells, resulting in a pristine decellularized organ matrix that can be stored for over 12 months. Next, the decellularized organ is placed into a bioreactor, and we initiate our patented recellularization process. During this step, human cells are introduced to the organ, and over the next 14 to 28 days, the organs are continuously monitored and perfused with the appropriate nutrients and oxygen to achieve the desired key growth parameters, resulting in a bioengineered organ. I always love that video. It summarizes up really fast some of what we're doing to get you up to speed. Our process is really broken down into two aspects. First is that decellularization, the ability to be able to remove all the cells from a scaffold. It was important early on that we demonstrated with this that we could start with a porcine organ, use perfusion decellularization to remove the cells, and demonstrate that we have complete viral clearance. We actually commercialized two products where we took full porcine livers, perfusion decellularized those, and created a surgical mesh called Miromesh that's been transplanted into thousands of patients. So we've got a really good safety profile associated with the matrix themselves. The next step in this, and I'll dive in a little bit deeper, is the recellularization. That's the ability now to be able to take that scaffold, introduce human cells in that to be able to bring that functional organ back. That's really how we bioengineer an organ, and allows us to get by a couple of the constraints or address some of the constraints in the field today, primarily around xenotransplantation in porcine viruses. We have already demonstrated that viral clearance, but more importantly is the ability to be able to scale this. If we look at transplant field in general, and some of the comparisons and differentiation in the field, we all know today, human to human, that's the gold standard. The challenge with that is while we're able to overcome the immunogenicity of those organs, it's still supply constraint. The second area of technology trying to address this tremendous need is xenotransplantation, and that's the ability to genetically modify the pig. Instead of taking that organ out and removing the cells, the notion is can you use genetic manipulation to be able to essentially make that organ be able to be transplanted from the pig directly into the human. A lot of news has been around this. We've seen some descendant studies on the kidney side, and certainly the heart transplant that occurred earlier this year with David Bennett that lasted 60 days, which was a great step forward for the field. I think what the outcomes from that and the challenge was, one was the porcine virus that was found in the recipient, as well as just the level of immunosuppression that's needed as you're looking at xenotransplantation as it moves forward. We really address both of those in the sense that we're starting with that decellularized matrix. As part of that matrix that I talked about a second ago, as part of our process, we've already validated that we have viral clearance. So we're not worried about, from a safety standpoint, of anything being exposed to the patient. And the second part that I'll get into a second is our ability then to be able to recellularize that scaffold with human cells. And that specifically also addresses the immunosuppression standpoint, where from our plan is to use immunosuppression that's similar to donated organs today. So if we dive into our process just a little bit more, the recellularization, once the organ's decellularized, we take that organ, place it into a bioreactor, and then we continually perfuse that. At that point, that's when we add in the human cells. That process takes about 2 to 4 weeks to continue to mature to the point that you then would have that functionality to be able to release that organ. A question we get a lot is, where are the cells coming from? So in the first or the current generation that we're doing today, we actually source organs that weren't placed for transplant. So whether that's cuts, nicks, tears, trauma, long ischemia time, that allows us to be able to work with organ procurement organizations to bring those organs into Miromatrix. And now we isolate out the primary cells. For the liver, we're isolating out the primary cells. For the kidney, we're isolating out the primary cells. We're then able to recellularize the organ with those primary cells. And the reason we're really doing that for the first standpoint is we're looking at how we fulfill our mission. How do we get organs to patients as fast as possible? So from a regulatory standpoint, we're approaching the first one with a scaffold that's already been cleared, and human primary cells, unmodified, that today is part of transplant, combining those together for our first products. And then once we're there and have that ability to be able to commercialize those, save lives, then it's our ability of the next generation then to focus on stem cells, where you can start to focus on that holy grail of transplant. How can you bioengineer an organ that no longer needs immunosuppression? Really doing it in a stepwise fashion. If you look at our facilities, we've really created a world-class manufacturing facility that we opened at the beginning of this year. Gives you a sense that you saw in the video, but also the sense here, the scale. We're able to bioengineer these in a closed system that's highly scalable. But more importantly, we've already invested in the infrastructure and have the manufacturing facility to complete our development, but also to be able to support our phase 1, phase 2, and phase 3 clinical studies. Just to give you a sense of the scale, we've already bioengineered over 400 organs in our facility since we opened that earlier this year. We've really always relied on a development path to de-risk our platform and bring that forward. And that really started with the first major milestone. We needed to demonstrate that the decellularization process itself was safe and scalable. And we really achieved that, as I mentioned in the beginning, by commercializing MiroMesh and MiroDerm. This is decellularized porcine livers. That allowed us to go forward under a 510K, manufacture that, demonstrate viral clearance. More importantly, thousands of patients have been treated with this product without any adverse events related to anything immune related. Our next step that we're on the cusp of, and I'll dive into this in a second, is to next pull together the decellularization, but also now demonstrate that we can effectively recellularize and bring that function back to the organ. And what we're focusing on that will be our first clinical trials will be what we call the MIRO liver ELAP, or the external liver assist product. That's where we're going to take the bioengineered liver and we house that external to patients with acute liver failure. That allows us to be able to treat those patients and demonstrate the effectiveness as well of a bioengineered liver. And then both of that is then sequential to moving forward our 2 lead products, which is our bioengineered liver and our bioengineered kidney, which would be fully transplantable. If we look at this in a pipeline standpoint, where this sits, today we've already filed our pre-IND and had a good meeting and dialogue with the FDA on the MIRO liver ELAP, and we're getting ready to file our IND by the end of this year on that product. And then on the MIRO kidney and MIRO liver, both of those, we plan to submit our pre-IND next year. So if we dive into the data a little bit and what excites us on this and where we are, certainly on the liver, not to go too deep into the market, but there's tremendous need here. I mean, if you look at liver failure, there are no drugs, there's no devices, there's no dialysis for liver patients. Today, it's really transplant, or unfortunately, you don't have access to that. So today, we're losing about 50,000 patients annually in the US alone because we don't have access to enough transplantable livers. So what we've already demonstrated and published on is our ability to revascularize the liver. So in this case, we took a liver graft, decellularize that, revascularize that with human endothelial cells, and now we transplant this back into a pig. We take the clamps off of this, and now you can see that initial perfusion of the graft, where you're going to see that initial inflow as we pull off the portal vein, and then it's still going to be white up on the top there. Once we pull off the vena cava, once we pull that clamp off right there, now you can see complete perfusion. So we really solved the vascular challenge here, because at this point we're able to anastomosis back into a recipient. We published on this in Nature Biomedical Engineering, demonstrate that we could get long-term perfusion. Once we solve that, we then focus on how do we demonstrate that we have liver function. So we recellularized with the hepatocytes as well. We can demonstrate that we have albumin, we can demonstrate urea. More importantly, we can demonstrate that we can dose in high doses of ammonia and we get beautiful clearance. You gotta remember, the liver is the one organ in the body, the only organ in the body that can do ammonia clearance. We've demonstrated many other functionalities as well, but these are some of the key ones. So once we had that functionality, we wanted to create an acute liver failure model. We published on this as well in Nature Communications Biology, where we created this acute liver failure model, essentially ligated out the native liver, and then transplanted in our bioengineered liver, and then looked at that. And you can see in the control, similar to what we see with acute liver failure patients, ammonia goes really, really high. At that point, those patients usually go into a coma, and then we lose them. What we were able to demonstrate with our bioengineered graft, in this case, which was about a third of the size of the native liver, is we were able to transplant this back in. Ammonia goes up, but starts to level off. In some cases, is coming back down. So now we're able to demonstrate what we saw on the bench. We're seeing in the large animal model as well. Since then, we've increased our liver graft size, increased the payload of hepatocytes and liver-specific cells in there. But demonstrating that in the acute liver failure model, this really sets us up for our first clinical trial that we have planned and are filing our IND on this shortly. And that's the MIRO liver ELAP. That's the ability to take the bioengineered liver, house that external to the patient, essentially create liver dialysis for the patient. But the liver is the one organ in the body that can regenerate itself. So what we're looking at this therapy is both a bridge to transplant, provide those patients a longer time and more support to potentially get life-saving technology, or also hopefully a bridge to recovery. But this really allows us, while this is a safety study, we'll still be able to have secondary endpoints to be able to look at the functionality of the bioengineered liver in this application. So this is a rapid study. This is up to 3 days of continuous therapy for these patients. So we're excited to be moving that forward. The other large program within Miromatrix is our kidney program. As you all can imagine, tremendous need here as well. We have about 550,000 patients in the US on dialysis today. We know that that's not a great solution because we lose 50% of those patients over 5 years. So we're looking, what is that alternative? We know with transplant today, of recipients who receive a kidney, the mortality rate associated with that is about 15% over 5 years compared to 50%, and the quality of life is superior. So we know by all means being able to have access to transplant would be life-changing for these patients. Real quick, just to demonstrate this, and if you're interested, go to our website. We have videos and other things to dive into this a little bit deeper. But this just shows our ability to be able to functionally recellularize the vasculature of the glomerulus, which is important for the filtration, as well as be able to demonstrate, similar to what we demonstrated inside the liver, is we can revascularize the kidney construct with human endothelial cells, transplant that back in, come back 7 days later, and we have complete patency inside the angiogram. So we solve the vascular challenge there as well. And then similarly, we can recellularize the kidney, transplant that back into acute models, and then start to look at effluent or early urine production that are coming off of these models as well. So we're continue to develop really good data on this. What we're looking and focused on right now is just further increasing the recellularization. We have the appropriate function that we want to see. We want to increase the density of that recellularization, where in these images we're about 30%. Today I'd say we're closer to 50%. And we're setting that threshold closer to 70% to move forward with our pre-IND and then utilize that data to move forward with the IND. So excited on where this is moving as well. Just a quick introduction to the company, further great management team, board of directors, key advisors who really bring all this together to be able to move this forward and look at how we help patients. So in summary, we are mission driven from the aspect of we want to eliminate the organ transplant waiting list. I mean, the market opportunity and the need is tremendous, with almost 120,000 patients today waiting for an organ. Kidney and liver make up 95% of that, which is really why those are our initial focus. What we can do with our technology, any organ inside the body, our focus is really where is the greatest need today. But the true need far exceeds those who are on the list, because today we can't deliver to the vast majority who are on the list and provide them a therapy. We got great technology and a proprietary two-step decellularization, recellularization, which really we feel addresses some of those true challenges that are facing xenotransplantation today. And more importantly, because we're not animal-derived, we are not a xenotransplantation from that classification. We've developed a great pipeline, really excited about where we're going, but more importantly, just excited about changing the future of transplant. So, thank you.